Lipids are macromolecules that are large organic molecules. They are insoluble in water but can dissolve in other lipids and organic solvents such as ethanol. They are made from the elements carbon, hydrogen and oxygen, with the proportion of oxygen to carbon and hydrogen smaller than in carbohydrates. There are 5 groups of lipid molecules. These are triglycerides, steroids, phospholipids, waxes and cholesterol. The central role of the lipid is in the plasma membrane, where phospholipids provide flexibility and transfer lipid-soluble substances across them. Other roles of lipids include in protection. Fat is stored around organs such as the kidneys, protecting them as they act as shock absorbers as these organs are vulnerable to knocks. An energy store. When oxidized, lipids for the same mass store 3 times as much energy as carbohydrates. Insulation. Beneath the body's surface, they can retain heat as they are slow conductors of heat. In winter, mammals tend to increase in their adipose tissue to reduce heat loss. Waterproofing. They are insoluble in water and act as a waterproofing material in plants and animals. In plants, the waxy cuticle is important for preventing water loss. And finally, they are crucial for buoyancy. Many single-celled aquatic organisms produce oil droplets to aid in buoyancy. Triglycerides can be referred to as fats or oils. Although chemically similar, fats such as butter are solid at room temperature, whereas oils are liquids. Fats come mainly from animal sources and oils are mostly from plant sources. Triglycerides are made from 1 glycerol and 3 fatty acids. The hydroxyl group on the glycerol undergoes a condensation reaction with the carboxyl group on the fatty acid. This forms 3 ester bonds and 3 water molecules are removed. These bonds can be broken down in hydrolysis reactions, which would produce glycerol and 3 fatty acids. The properties that different fats or oils shows comes from the variation in the fatty acids, as the glycerol molecule is the same. Fatty acids may be unsaturated or saturated. The carbon-to-carbon atoms contain double bonds in an unsaturated molecule, such as in oleic acid. In fatty acids that have 1 carbon-to-carbon double bond, they are called monounsaturated fatty acids. Those with 2 or more carbon-to-carbon double bonds are called polyunsaturated fatty acids. The arrangement of molecules around the carbon-to-carbon bond can differ. This arrangement can either be in a cis or trans form. In the cis form, the presence of the double bond creates a bend in the chain. The more double bonds, the more bends. This makes it difficult for them to pack closely together, and also explains why they're liquids at room temperature, as the bonds between the chains is weaker. In the trans form, however, the chains tend to be straighter and therefore resemble properties seen in saturated fatty acids. In saturated fatty acids, each carbon atom in the hydrogen-carbon chain is linked to a carbon atom on either side and also to 2 hydrogen atoms. This means that the carbon atoms have the maximum number of bonds. Therefore, saturated fatty acids only have single carbon-to-carbon bonds. This makes the chain straight, allowing them to pack closely together, which is why they are solid at room temperature. Phospholipids form an essential part of the cell membrane, forming the biolayer structure. They have a similar structure to triglycerides, but the 3rd hydroxyl group on the glycerol molecule is bonded to a polar phosphate rather than a fatty acid. We describe these molecules as amphipathic, where the polar phosphate group is strongly attracted to water, which we call hydrophilic. The fatty acid chains are non-polar and therefore repel water, which we call hydrophobic. This means that when phospholipids are placed in water, they can orient themselves by either forming a monolayer where the hydrophilic heads are facing into the water and the hydrophobic tails away from water, or a bilayer where the hydrophobic tails again are facing away from the water. There is another way in which phospholipids interact with water, which we can see in the micellar structure. A micellar consists of a single layer of fatty acids arranged in a globular fashion. The polar heads point towards the outside and interact with the aqueous environment, while the hydrophobic tails are packed into the inner space and are prevented from interacting with the aqueous environment. The composition of phospholipids contributes to the fluidity of the cell membrane. If they are mainly saturated fatty acid tails, then the membrane will be less fluid. If they are mainly unsaturated fatty acid tails, then the membrane will be more fluid. If the membrane is too fluid, it will let more molecules
through it, and if it is not fluid, it will prevent molecules from passing through it. Phospholipids control membrane protein orientation. Weak hydrophobic interactions between the phospholipids and the membrane proteins hold the proteins within the membrane, but still allow them to move within the layer.
Aside from phospholipids and triglyceride, other lipids include steroids, cholesterol, and waxes. Cholesterol is vital in the manufacture of vitamin D and has a role in the cell membrane.
Just like phospholipids, cholesterol is an amphipathic molecule having both hydrophilic and hydrophobic regions. Cholesterol's hydroxyl group is hydrophilic and aligns towards the phosphate heads of the phospholipids. The steroid ring and the hydrocarbon tail is hydrophobic and associates with the phospholipid tails. Cholesterol affects the fluidity and permeability of the cell membrane. As we mentioned before, if the membrane is too fluid, it will let more molecules through it, and if it is not fluid, it will prevent molecules from passing through. Cholesterol disrupts the close packing of the phospholipids, increasing the flexibility of the membrane, preventing it from stiffening or crystallization. As the concentration of cholesterol increases, so does the fluidity. It also does something very interesting at different temperatures. At high temperatures, cholesterol helps reduce fluidity, and at low temperatures, it increases the fluidity. In this way, it has an important role protecting membrane fluidity from fluctuations in temperature. It also acts as a barrier, fitting in the spaces between phospholipids, preventing water-soluble substances from diffusing across the membrane.
Steroids contain a ring structure. In animals, many hormones are steroids, including testosterone and estrogen. These are also made from cholesterol. Waxes have a similar structure to triglycerides but have fatty acids bonded to long alcohols rather than glycerol. At room temperature, they are usually solids and provide protection and waterproofing on the surface of leaves and insects. Hi, my name is Mr. Science, aka Salim. If you're new to the channel, please remember to like and subscribe, and for more teaching resources, you can visit my website at www.mrscience.co.uk.